It's a little bit overwhelming. When we've had some of us in the room remember the drought where we weren't finding results and it was very frustrating and grants weren't getting funded. And now we have so much data and so many research questions, and we're needing to develop new bespoke methods to really understand it.
Yeah.
And really great to welcome Gerome Breen as our next speaker. Gerome and some of you in the audience have attended a conference in Copenhagen just earlier this week, sponsored by Lundbeck and iPsych, talking about leveraging the discoveries from genetic studies into discovery of new products, new treatments. So it is a very exciting time, and the field is moving so quickly, it's actually hard to keep up with. So Gerome is a reader of neuropsychiatric and translational genetics at King's College London. He's a psychiatric geneticist, very interested in mood disorders, psychosis, eating disorders, and he's a key member, as Naomi is, of the incredibly productive Psychiatric Genomics Consortium, or PGC. And this has been one of the great sociological success experiments in science, where many people from around the world have combined data in a very open way, and we've really benefited from that. And Gerome was involved in the paper that Naomi showed, the landmark Nature Genetics paper, which we just need to pause and reflect that this is a really important paper looking at the genetic architecture of depression. And Jerome's going to follow that through the genetics of mood spectrum, looking at 600,000 individuals. Gerome, we look forward to your presentation. Thank you.
So I don't know how I'm going to follow Naomi's talk, which I thought was wonderfully clear. I'm probably going to end up confusing you. So my abstract is basically a pure GWAS, but having seen yesterday, I decided to introduce some epidemiology. So just one thing to reflect on is that in genetics, it's not just schizophrenia that has had successful GWAS now in psychiatry. It's virtually every psychiatric disorder. Once sample sizes get significantly beyond 20,000 samples in almost every psychiatric disorder, we tend to see sort of good discovery. But notably, only 2 disorders are in the linear phase of discovery. That is, we can predict how many loci that we'll discover. So in schizophrenia, roughly every 300 to 400 cases that get added, you get another GWAS hit. In depression, about 1 every 2,000. So I have, this is my current tally of the GWAS hits, and I'm going to present you now. So it's 44 was published in Nature Genetics just shortly. We've added another set of cases to that, and now we have 55 in a fairly predictable manner. Okay, and AN is anorexia, by the way, so that's got 8 now as well. So first of all, just to give you some background, so UK Biobank is a sample on which a lot of us work. It's 500,000 UK adults, aged between 40 and 70 at recruitment, with a wide range of baseline phenotyping. About 300,000 people are recontactable for additional phenotyping online, and the study is not very ethnically diverse, but it does reflect the UK population of that age. It's slightly less socioeconomically deprived than the UK average, but actually it's pretty representative overall. The UK Biobank is an ongoing study with data being generated all the time. So last year we had the release of the genotyping data and imputed data for 500,000 individuals, so genome-wide genotyping. We also had then the release of an online mental health questionnaire, actually in 157,000 people. And there are ongoing studies in UK Biobank, such as neuroimaging of 100,000 people, whole genome sequencing of 50,000 people, exome sequencing of the whole cohort. So it's going to become a very large contributor to our studies going forward. The other notable thing which is going to happen this year is electronic medical record linkage to primary care records for about 3/4 of the data set, actually. So that will be quite useful. So the mental health questionnaire was processed. So this slide outlines the process, and just say that it was relatively straightforward, actually, for a project of this size. The mental health questionnaire content essentially it's largely based off a CD short form core of questions focused on depression and anxiety, supplemented then by recognized instruments such as the GAD-7, PHQ-9, the audit, and a set of questions, around 26, that address life events, so childhood trauma, the adult versions of childhood trauma, and catastrophic life events. These are the ends from the mental health questionnaire. So of the 67,000 people who ended up answering the depression and anxiety questions, we have about 30,000 people that meet the criteria for a diagnosis of depressed ever. So for example, there were 17,000 people with apparently recurrent depression, almost 12,000 people with single episode depression. So before we go on to do case control analysis, one of the things about having such a large data set all answering the same questionnaire is that we don't have to then go to the diagnostic categories, at least first. We can do GWASs of the actual questions themselves. And one thing that Naomi brought up is basically that nearly every question you can ask of a human, or nearly every phenotype you can measure, has a distinct genetic and environmental component of its own. So the question really is not G by E, right? It's in genetic terms ACE by ACE, so genetic and environment by genetic and environment by genetic and environment. So investigating that is complex, and I'll come on to that later, but genetic correlations have some nice structures, nice properties that phenotypic correlations don't. Namely, you don't need people to answer the same questions. So if we do GWASs of all of the questions in the mental health questionnaire, can we find what symptoms genetically cluster together? And can we find potential genetic subtypes of disorders? So we did a symptom level GWAS in the MHQ. So mental health questionnaire, we dichotomized questions where they were categorical, and we did quantitative GWAS where it was quantitative. We used standard approaches to GWAS in UK Biobank, and then we calculated genetic correlations between the summary statistics for all the questions. We then carried out hierarchical clustering of all questions and of subsets of related questions. I'm not going to show all of that data. I'm just going to show, well, I'm showing all of that data. These are the correlations between all of the questions in the MHQ. Now, what you can see here is that the clusters aren't very distinct from one another. There's a lot of off-diagonal correlation. But roughly speaking, up here we have lifetime depression and anxiety clustering together, as you might expect. Here we have life events. These are reported traumatic life events and catastrophic life events having a heritability and clustering together. This is recent anxiety, depression, and PTSD clustering together. Now the off-diagonals are significant, so the recent anxiety and depression has a reasonably strong correlation with the lifetime anxiety and depression. But nevertheless, it does cluster on its own. There's a well-being section to the questionnaire, which thanks to the way the questions are asked, has negative genetic correlations with these other questions, but it does cluster separately. And addiction, notably, clusters on its own, relatively on its own, right? So there's still off-diagonal correlation, but it does seem to come out fairly distinctly. So but I think one item here though is this cluster around life events. So you can see that they do have some genetics. And so you can see that more clearly if you do this as a dendrogram. So over here, so these are the boxes. Boxes outline what's in each thing. So here we have recent anxiety and depression. Here we have lifetime anxiety and depression, and here we have life events, sitting essentially on the same part of the tree. And that leads me on to a study that we've been doing, Naomi mentioned, on trauma and depression in UK Biobank. So trauma in UK Biobank was measured by a short form of the Childhood Trauma Questionnaire, and we assessed then, and an adult version of those questions and traumatic life events. And we assessed the association of those phenotypically with depression. And we said we were interested in life events with an odds ratio over 2, in particular to construct our strata. Felt loved as a child, had a very strong effect. Felt hated by a family member, had a very strong effect. Sexually abused, had a very strong effect as well. Although very comparable to the, say, felt hated and felt loved. The adult questions, hit by a partner, belittled by a partner, and sexually assaulted by a partner came out very strongly. But again, a bit like the felt loved as a child, the belittled by partners seemed to be having quite a very strong effect. In catastrophic life events, although all of these are, of course, massively significant, sexually assaulted had by far and away the strongest association. We defined then a trauma-exposed group as having 2 or more, answering to 2 or more of these very strongly associated life events. That gave us then case and controls of the following. So we had about 13,000 exposed cases and about 9,500 unexposed cases. We had 10,000 odd exposed controls and nearly 40,000 unexposed controls. First of all, we just did straight GWASs of depression cases versus controls. This was the GWAS of depression. We had a reasonable SNP heritability towards 20%, but we didn't have much GWAS signal consistent with other GWASs of depression at this sample size. This is a PGC hit, probably. It's also a neuroticism hit and an anxiety hit. However, reported trauma exposure by itself also had GWAS hits. In fact, it had 6 GWAS hits. And it had a significant similar heritability in these analyses. So, key questions we wanted to ask of the data, some of which we were able to answer and some of which we were not, were: is trauma-exposed depression more heritable? Is non-trauma exposed depression more heritable, or is trauma-exposed depression more heritable? So we started to look at this and we got some interesting results. So we assessed the SNP heritability of the strata of depression, And here we looked at the heritability of trauma-exposed depression at a range of population prevalences, because we were a bit uncertain about what prevalence to adopt, but probably around here somewhere. And at first glance, it looked like the exposed group had a higher heritability than the unexposed group. However, I was thinking about having Naomi's head here, but I decided that would not be good. So after several discussions with Naomi and Wouter, then we came to the conclusion that the problem is here in this data set is that depression and trauma also share a genetic basis. So depression and trauma have a significant genetic correlation in the full cohort of around 0.62. Even if you look at reported trauma exposure in control, so people who do not endorse depression or anxiety, the reporting of the trauma exposure has a genetic correlation with MDD of 0.31. So that meant that assessing, when you've got a genetic correlation between things, assessing differences in heritability between strata is invalid, essentially. But the genetic correlation method, however, is robust to this type of confounding. So we can ask instead a different question. Does exposed depression show a different set of genetic correlations to unexposed depression? And so first of all, we looked at the genetic correlations with psychiatric disorders and traits. And the take-home message here is that trauma-exposed and unexposed depression has the same set of genetic correlations with psychiatric disorders and traits, such as neuroticism. However, when you look at genetic correlations with physical health measures, these differ by trauma exposure in depression cases. So the genetic correlations with, for example, body mass index and depression overall are just about significant here, about 0.11. But when you stratify by trauma exposure, you can see here that in the trauma exposed group, it's 0.21, and it becomes non-significant in the trauma unexposed group. And this pattern repeats for waist circumference and waist to hip ratio. However, we also had this genetic correlation, which is a little harder to explain, which is femoral neck bone mineral density. So hip bone mineral density. And that has strong negative genetic correlation in trauma unexposed depression. It's worthwhile noting that in bipolar disorder, we do see somewhat similar genetic correlation for bone mineral density, although it doesn't pass multiple testing correction. This may or may not be related, but there are many, many calcium channel genes in the genes that we find to be associated with psychiatric disorders overall. We then turn to doing a polygenic risk score by trauma interaction. So you can test an additive interaction using linear regression. You can test a multiplicative interaction using logistic regression. The interpretation is difficult, and there are papers on the topic by Wouter as well. One of the complications is that you've got to use full models. So Matt Keller had this paper a few years ago saying you should have interaction terms for all your covariates. So you should use a full model. If you use the full model, some methods that have been used in the field, like RERI, for example, become unstable. So that's why linear regression is used. So what we observed then was a significant additive interaction term for linear regression. So that is the combined effect of the polygenic score for depression and reported trauma exposure was greater than the sum of their individual effects. And there was no significant multiplicative interaction term observed. So the combined effect of the polygenic risk score for depression and reported trauma exposure was not greater than the product of the individual effects. conclusion from this then is that genetic risk for depression and trauma exposure combine in a multiplicative rather than an additive way to influence depression risk. BMI was also significant, but it was neither. It didn't conform to either a multiplicative or an additive model. It was a bit more complex. So depression and trauma are difficult to disentangle. So just one thing to say here, right, is that because you have a genetic correlation between depression and trauma, doing gene environment studies is fundamentally difficult. There are some studies out there like Converge, which is the study of Chinese very severe depression, which got very different results to the ones I've just presented, but they have a very different sample size as well, so a rather smaller sample size. How am I doing for time? Yeah. Okay. So, but the genetic correlations are very interesting. And the narrower range of genetic correlations in trauma-exposed depression and the lack of correlations in trauma-unexposed depression and the lack of correlation with BMI does seem to echo earlier ideas of endogenous depression. Okay, so next, just some very, very brief things. So that's very, very complex and very hard to interpret. So let's go back to doing simple GWAS. So I'll just go forward. So this is the paper that came out. This is the Manhattan plot from the paper that came out in Nature Genetics a few weeks ago. 44 hits for depression, 135,000 cases, 344,000 controls. There's also a paper under review in Nature Genetics from the PGC bipolar group, which reports 30 loci with a replication effort, but this is the Manhattan plot with 19 GWAS loci. We put this together with the depression cases from UK Biobank, and then conducted a meta-analysis of that. So we have about 600,000 individuals in this meta-analysis overall. And what we discovered then when we meta-analyze everything together is that we have 73 loci for mood disorders. 4 of the 19 bipolar loci came through this meta-analysis, and 39 of the 44 MDD loci came through. But there are 30 new loci overall in this analysis. I should say that in our depression alone analysis, we had 55 loci. So revisiting the GSMR results that Naomi presented earlier, we had some additional power to test things. And I won't go into all of this, but what we did see was there appeared to be, so Naomi doesn't like the word appear, but the evidence was consistent with a causal effect of MDD on coronary artery disease, but not in the other direction. The only other point I'd like to make is that IQ and psychiatric disorders in general, IQ and depression, bipolar, do not have any genetic correlation, and there doesn't appear to be any causal path between them. But educational attainment is interesting. It seems to have a bidirectional relationship, and if you look at it in, whereas if you look at for MDD, whereas if you look at it in bipolar, there seems to be a relationship from educational attainment to bipolar. Okay, so I think that's my final slide then. So there's 2 things. So everything has a G and an E, but 2 things I want you to take away is that we can do some causal epidemiology. Coronary artery disease is looking interesting. And the new meme is that MDD is the second most successful psychiatric disorder for discovery. So no more that depression genetics doesn't work. Okay.
Thank you. Thanks, Gerome, and congratulations to the PGC for this fantastic work. And I really appreciate that you're sharing these new discoveries with us, and I'll ask the audience to respect not tweeting some of that new material. That finding about IQ is fascinating. I thought that IQ was linking to all mental disorders, but not depression. So.
No, I don't. I think so. The genetics of IQ do not appear to be important for most psychiatric disorders, but educational attainment.
Oh, yeah, okay. I keep getting this confused.
But the educational attainment, it seems to be a point of distinction. Educational attainment GWASs appear to have some relationship. So there's some, but IQ and educational attainment are highly correlated things, but they're not the same.
Okay, first question from Andrea.
Hi, I have two questions. The first one is, you showed the heat map of the correlation from a genetic perspective. If you do the same exercise from a phenotypic perspective and you compare the two, do you have any surprise, or you see just replicating all the genetic correlation replicate the phenotypic correlation?
Yeah, doing this exactly the same analysis, because there's tree branching logic in the questionnaire. So even if you answer the screening question, there's tree branching logic, so not everyone answers the same questions. So you get this apparent, if you try to do it with methods that allow for that, you get this apparent lack of off-diagonal correlation.
Right, okay.
But it's because not everyone is answering the same questions, I think. But the phenotypic correlations are more clear-cut than the genetic correlations overall, I would say, just from what we've looked at.
Yeah, and related to the question, it seems that there's a lot of redundancy in those questions, right? Some of them, they're highly genetically correlated. Does it tell you something about designing questionnaire for future study, how to optimize that in a way that you kind of, capture truly independent component rather than asking 3 questions that really capture the same thing?
Yes, it does, and we've thought about that, and so we're actually launching another large study to do recruitment at the moment, and we're taking some of those things into account. Yeah.
Other questions? If not, we will, before we thank Gerome, we'll just to remind you we have a half an hour break now, or we'll be back at 11:30 for a really important address from Peter Vischer. I think we're going to have the morning tea outside, Agneta and Sina, is that all? We've put the tables outside, and there are more mugs to be given out, coffee mugs this afternoon, if you want to get your official Niels Bohr professorship mug. But we've had a fantastic start today. I'm really grateful to Naomi and Gerome for really masterclass in psychiatric genetics. And I really want to, from the bottom of my heart, congratulate you both for the impact you're making on the field. So please join with me and thank Gerome and Naomi.